often think about cellular membranes as being these greasy lipids, but in reality, about half of the mass of the membrane can actually be proteins. And proteins that are embedded in this membrane make up about 60% of pharmaceutical drug targets. Not just because they're kind of for drugs to bump into, but also because they can play key roles serving as receptors that trigger downstream pathways, and therefore, if you tweak them, you can kind of tweak a fire hose and then have big effects downstream. So lots of cool stuff. But membrane proteins are also difficult to study because at least parts of some of them are inside out, kind of. Let me explain what I mean.
First, just to note that I'm going to be talking mostly about integral membrane proteins. So there are two main classes of membrane proteins, and exact classification is going to be vary depending on where you're looking. But we can classify them as being integral membrane proteins, or IMPs, if they're embedded in the membrane, at least partly. And so as we'll see, many of these are actually going to pass through or transverse the membrane. They're going to be transmembrane proteins. And then we have peripheral proteins, which are basically, they're kind of like reversibly stuck on one side of the membrane. They might be anchored on there by a connection through to one of those lipids, or they might just be weakly bound either to the heads of those lipids, say, or to integral membrane proteins that are already there. But we're going to focus on these integral membrane proteins for the most part. And when I talk about them being inside out, we need to think about what their structure is like. And what their structure is like is going to be dictated by what they're made up of and the environment that they're in. So when we think about like a typical soluble protein, when we talk about something being soluble, it means it's surrounded in the coat of the solvent. And so normally when we're thinking in biochemistry, we're thinking about proteins being in a watery or aqueous environment. This could be the inside of a cell, so your cytoplasm, or this could be outside of a cell, the extracellular environment. Both of these are going to be aqueous. They're going to be all watery. And so proteins that are happy in those environments are going to have, at least on their outside, they're going to have amino acids, so they're going to have protein building blocks that are going to like water. They're going to be hydrophilic, and water is going to like it. So it's going to hang out with it and it all is going to be good. Now, these proteins are also going to have regions that are hydrophobic, but those hydrophobic or water avoided regions are going to be clustered in the center of the protein. And this is a key driving force in how proteins fold, is going to be clustering up those hydrophobic parts in the inside of the protein and the hydrophilic parts on the outside. And that works great if you're out in a watery environment. But when we're talking about a integral membrane protein, part of it at least is going to be facing a lipid environment. So it's going to be facing like a kind of anti-water environment, so like an all greasy environment. Because if you think about our cellular membranes, they're made up of phospholipids, a phospholipid bilayer. And phospholipids are what we call amphiphilic. They have both hydrophobic parts and hydrophilic parts. Their hydrophilic parts are going to be these water-loving heads, and then they're going to have these hydrophobic or lipidy tails. And this is going to make them form these lipid bilayers. And if you have a protein that has to go through that lipid, then that protein is going to have in that transmembrane domain, it's going to kind of have to be inside out. It's going to have its in hydrophobic parts on the outside and its hydrophilic parts on the inside. As we'll get into, this is going to make it hard to study them because we typically like to study things in a watery environment. And if we're going to want to study these full-length proteins, we're going to have to stabilize that transmembrane domain outside of its normal home. And we'll see how we can do this, such as with detergents. Membrane proteins play important functions, including serving as receptors. So they're going to be able to help sense what's going on so that the cells can respond to signals. These signals can be like chemical messages, various chemicals, hormones, etc. And what happens is if you have these receptors that are transversing the membrane, they can bind to those chemical messengers and kind of change shape, undergo a conformational change. And by doing this, they can relay the signal to the inside of the cells. Now, often what happens is you get a signaling cascade. And so if you have a receptor and you impact the receptor, you can impact a lot of downstream things. So not only is that receptor easier for the drug to find than if something was buried inside of a cell, but it can also have dramatic effects because that receptor is going to pass that message on. And so the thing that the receptor binds is called the ligand, and a ligand can be an agonist, it can activate that receptor, or what many drugs are antagonists, where they bind to and prevent the activation of the receptor, and therefore prevent the downstream signaling. When we talk about downstream signaling, sometimes the downstream signaling comes from the receptor doing something like phosphorylating a protein. So it will put a phosphate group, this charged group, on a protein, and that protein can then be activated, say, and go send the message further on. Other receptors, they actually work in hand with the signal relayer, something such as a G protein. So we have these type of receptors called G protein coupled receptors. And what happens is you have this integral membrane protein, this G protein coupled receptor, and it's going to bind to the message, but it's also bound to this little peripheral membrane protein called a G protein. And then the signal, when this integral membrane protein gets the signal, it kind of passes it on to that G protein, which then moves away from the receptor and goes and passes on the message. So in this case, you have two membrane proteins playing a role in helping relay this message. Speaking of like teamwork, membrane proteins can serve as scaffolds, which kind of help tie together other proteins. So when you have a reaction that you want to happen in a cell, say, or you have some sort of pathway, if you think about a car assembly line, there's a lot of different pieces that have to all be brought together in the same place. You have to have a bunch of different robots passing one thing off to another, etc. And if those robots can't find each other, it's going to be hard. And so if they're just like wandering around the factory, that's not going to be very useful. And so by scaffolding the molecules together, we can kind of membrane protein complexes are able to make it so that reactions and things like this can be more efficient. And so often you'll see, such as in the mitochondria, where you have the ATP generating machinery, all of that can kind of cluster together in order to make reactions happen faster. And with signaling cascades, it's going to be important to scaffold things together as well. Membrane proteins don't only serve as passing the message indirectly, they can also kind of just let things pass through. So transporters are membrane proteins that traverse the membrane and they allow things to get through. This can be active transport, which is going to require energy, such as through ATP, or it can be passive. So if you think of something just like a pore, but not just like a total pore, it'll have some selectivity. So it'll only let certain things clip through. A key example of these are like porins. So there's like aquaporin, which lets water through, various other transporters that let things in and out of the cell or compartments in the cell. Because although we typically think about just in terms of the outer cellular membrane, maybe the plasma membrane, we have to remember that there are also organelles inside of cells. So membrane bound compartments, things like the nucleus, the mitochondria, we have lysosomes, where things kind of get degraded. The ER, the Golgi, all these various membrane bound compartments inside the cell. And all of these have slightly different lipid compositions and different proteins that are embedded in them. But when we're talking about the surface of the cell, the plasma membrane is what we're talking about. And in this plasma membrane, we could have embedded various membrane proteins that are going to allow the cell to adhere to other cells, as well as to keep them like attached to a dish, say in the lab. So they can adhere to substrates, so surfaces, as well as to other cells and things like this. Membrane proteins are often glycosylated, so they have sugar chains attached to them. And this can play various roles. It can do things like allow the cells some of those adhesion properties. It can also serve important immune roles, both positively and negatively. So often viruses, what they do is they actually there's membrane proteins, such as the ones that they use to dock onto cells, such as the coronavirus spike protein. they're actually going to be glycosylated, and this can kind of hide the protein surface from the immune system and make it harder to recognize. But the glycan chains can also play really important roles in human physiology, so in our own body health. So membrane proteins are really important, but they're going to be hard to study because of that whole inside out thing. Sometimes what scientists will do is they will just study part of the protein. So they'll just study one of the parts that's in contact with the liquidy environment. So either the ectodomain, so the part that contacts the outside, or the endodomain, the part that contacts the inside, and kind of like avoid this transmembrane domain. And this works fine if you just want to study interactions that are happening, say the binding of something up here. So you might study the receptor binding domain purified by itself. Or if you want to study what it interacts with on the other side of things, you can have the endodomain. And so these are easier to work with. But when scientists want to study the full-length protein, this is going to be more challenging. When you go and you purify a protein, typically what you're doing is you're trying to purify a soluble protein, one of those cytosolic proteins, these water-soluble proteins. But when it comes to a membrane protein, well, it's kind of soluble in lipid, meaning that it gets surrounded by lipid. And so that's what its normal solvent is lipids, not this water. And so what's going to happen is that when you break open cells, the things that are associated with the membrane are going to stay stuck in the membrane, and the things that are not associated with the membrane are going to be, and are happy, are going to be these water-soluble membranes that are going to stay in the liquidy part. And so normally what we do when we purify proteins is we lyse them cells. So we break open the cells and then we centrifuge them really fast in order to pellet out that membrane bits, as well as like insoluble stuff or in water soluble stuff. And that's going to pellet out. And then we just kind of like discard the pellet and work with the watery stuff, the supernatant, the liquid that's above it. But when we want to study that membrane protein, well, now don't throw away that pellet. That's where our protein is. And so I actually, in grad school, I did a rotation. So in a lab where I was studying membrane proteins. And so I had to always remember not to throw away that pellet because that's where my protein is. But how do I rescue my protein? There are different ways to study and purify membrane proteins for various purposes. And so if we remember, we have this embedded in the membrane, and normally it's used to being embedded in that membrane. And so if we want to study it, we need to keep it embedded in some sort of lipid environment. But we don't want all of that lipid environment that this protein was in. We don't want the entire membrane with all that other stuff. We want to purify our protein. So we need to extract it out of the membrane it's in into a kind of simpler, purer membrane. A common way that this is done is using detergent. And so we'll talk more later in a second about how this is actually the strategy used to purify the full-length coronavirus spike protein. So that protein that the coronavirus uses to dock onto cells and get inside in order to study in the lab, or to use it, make a vaccine out of it, such as Novavax vaccine. Scientists use detergent in order to extract it from the membrane. And detergents often used in the lab for extracting all sorts of membrane proteins. Why this works is detergents are these like artificial soaps. They kind of look similar to a phospholipid, so to the things that are making up this bilipid membrane, this lipid bilayer, but they're different. So phospholipids, phospholipids and detergents are both amphiphilic, so they have hydrophobic or water-avoided parts and hydrophilic or water-loved parts. With a phospholipid, you kind of have these two lipid tails. With a detergent, you only have a single tail. And the consequence of this is that phospholipids are going to form bilayers when you put them in water to avoid water, to have their hydrophobic parts clustered together in the center and hydrophilic parts in the outside, kind of like a sandwich. But with a detergent, what you actually get is something called a micelle. So these are more like cone shaped, whereas these are more rectangular. And so a phospholipid wouldn't be able to form a micelle very nicely. Instead, if it forms one of those bubbly type things, it'll be with a lipid bilayer. But with the detergents, you'll have this conical shape. It can nicely form these micelles. And what you can do is because it's kind of similar looking to those phospholipids, and it has that amphiphilic character, it can break up the phospholipid membranes. But then because it has this tendency to form micelles instead of forming these bilayers, it's going to actually form these micelles if you have enough of it. And those micelles are going to have your protein incorporated into them. And so with the Novavax vaccine, this is the strategy that they use, is using these detergents to extract the coronavirus spike protein, which they express from insect cells. And so then the insect cells make this protein and it gets embedded in the insect cell membrane. And then they purify it out of the membrane and make a vaccine out of it so that our immune system can learn to recognize it. And basically, they also have like an adjuvant in it, so something to make it so that your immune system reacts more. And you get something like this, where you get these like rosettes, where you have these micelles with a bunch of different spike proteins embedded in them. And I have a whole post on this if you want to learn more. Detergents are great for some things, but not so great for other things. And if one of the reasons is because you have that single lipid rather than a bilayer. And so sometimes, such as with structural biology, where scientists want to study the molecular shapes of molecules, they want something a little more realistic. They want something with a bilayer. And one of the things that they can use is going to be lipid nanodiscs, which are kind of like lifesavers around these proteins, these kind of disc-like molecules that form around these proteins. And I was actually in my rotation, I was trying to form these nanodiscs. Yeah, I didn't have very much luck, but scientists that do this a lot are really good at it. And so they can use these nanodiscs to purify these complexes surrounded in a lipid bilayer that they can then use for structural studies. Speaking of structural studies, for big complexes, they can even use a technique called cryotomography, where basically they look at the membrane as is, rather than looking at purified complexes. So tomography is becoming like a really big thing. So including, I mean, like single molecule cryo-EM is still a huge thing too, where you're looking at purified proteins. But for bigger membrane complexes and things like this, cryotomography is kind of like you look at slices of the membrane rather than looking at single particles that are suspended in this liquid in your grid, and much more on cryo-EM and other posts. When we're on the topic of structure, I'll get back to what I promised you I'd tell you more about, which is basically the structure of how these integral membrane proteins are able to traverse the membrane. I'm going to be showing you some figures from this Principles of Biochemistry, the Leninger Principles of Biochemistry. My favorite biochemistry book. It's the one I learned on, and I still love it. I've been like reading it this morning and catching up. We're not catching up, but reviewing. So basically, when we have integral membrane proteins, we can classify them further based on the number of passes. So how many times do they go through or traverse the membrane? And which side are the termini on? So proteins have two ends. They have an N terminus, which is like the start, and then they have a C terminus, which is like the end. And when you have something pass through a membrane an odd number of times, those start and the stop are going to be on opposite sides. If they pass through an even number of times, they're going to be on the same side. But that side can be either on the inside, such as in the cytoplasm, or on the outside, such as in the extracellular environment. And they can also be passing through different numbers of times. They can pass through and have an anchor, so they can be attached to that protein, to that lipid, through such as a covalent linkage, through a lipid, all sorts of various things. And so there are different ways that these are classified. Oh, they can also be made up of multiple different proteins. And so there's a lot of different classifications, and the classifications are actually going to vary depending on where you look. Speaking of where you look, I had mentioned before that about a quarter of our protein coding genes are thought to be recipes for membrane proteins. And how this is predicted is based on something called the hydropathy index, which can be used to predict transmembrane domains. So basically, if you think about what you would expect to see in a membrane protein, what characteristics could you see from the amino acid sequence? What you would be looking for is you want to see hydrophobic amino acids. So those amino acids that are going to be happy in a transmembrane domain. And the hydropathy index is going to basically tell you that. So what you can do is you can go along the length of a protein. And so the length of this, so this would be like the first amino acid, second, third, fourth, whatever, all the way down the protein. And then you look and see how hydrophobic is it, how hydrophilic is it? And you give us something called a hydropathy index. Basically, what this tells you is how mad or happy would the protein be, or the amino acid be, if you were to move it from a hydrophobic environment, such as inside of the membrane, to a hydrophilic environment, such as in the cytosol. And what happens is that what it's measuring is going to be the delta G, the change in free energy. And much more on this in other posts, but when something has a higher G, so a higher energy, it's going to be less stable. And so if you're talking about a positive delta G, you're talking about something going from a low energy, stable, happy state to a high energy, unstable state. And so this is going to be unfavorable. It's like pushing a ball up the hill. And so if you see a positive hydropathy index, this is telling you it would be unfavorable. So this is telling you it would be unfavorable to take these amino acids and stick them from a hydrophobic environment into a hydrophilic environment. So if you see something where you have this positive line above, so you have a line above the axis, this is going to tell you something is hydrophobic, and below will be hydrophilic. So if you look for a transmembrane domain, what you're going to see is going to see something that's, you have this cluster of you have this stretch of amino acids that are going to be hydrophobic. And if you have multiple passes, you're going to see multiple different peaks that are connected by kind of like hydrophilic parts, where you have the outside where it kind of has to change direction and things like this, as well as the ends. Now, more specifically, what you're going to see is you're probably looking for stretches of about 20 to 30 amino acids. And why this is because that's how many amino acids it takes to go through the membrane as an alpha helix. And often this is what's happening, is that you are going through the membrane as an alpha helix in a transmembrane domain. And those amino acids that are going through, each of them is going to have contact with the lipids, and so each of those needs to be hydrophobic. And because they're kind of in that spring shape, there's going to have to take 20 to 30 amino acids to go through the membrane, even though the linear distance isn't that long. Often membrane proteins have about 7 or so passes, and between each of those passes is going to be one of those hydrophilic parts that can then hang out on the outside, so you're able to change direction. As well as you might have hydrophilic parts on the ends, where you have the C and the N terminus in contact with the watery environment. So this characteristics, kind of like signature of this 20 to 30 amino acid, hydrophobic amino acids in a row, is going to hint that you have a transmembrane protein, and this is how scientists can then get that quarter of your genes being for membrane proteins, or a quarter of your protein coding genes being for membrane proteins estimate. It's harder to predict whether something will go through a membrane as a beta strand, however. As a beta strand, so one of those like kind of accordion style things, basically in this form, the protein is going to be in a zigzag that's going to be more extended rather than tightly coiled. And so it's only going to take about 7 amino acids to go through a pass. And only every other one of them is actually going to have to be in contact with the lipid. So every other one has to be hydrophobic, but the others can be hydrophilic, and this can be used to form a hydrophilic pore. And so here are some examples. I know I focus mostly on integral membrane proteins, but peripheral membrane proteins are really cool too. And so because they have this reversible interaction with the membrane, cells can kind of regulate when they come and go, and they can say, hold on to proteins until pre-make them, hold them onto the membrane, and then when they want to do something, they can say cut that linker off or something like that, release the proteins, and have some sort of function. So peripheral membrane proteins are really, really cool too. But when we think about a membrane protein, we're often thinking about integral proteins. And remember that cell membranes aren't just lipids. So about half of that mass of some of your membranes are going to come from proteins. So when I say some, it's about 50% for the typical plasma membrane. So that membrane that's separating your cellular interior, so your cytoplasm, from the extracellular environment. It gets up to about 75% or so for some mitochondrial membranes, where you have that energy making taking place. It takes a lot of protein machinery and it's kind of all clustered together. Since lipids are much smaller than proteins, though, there's actually about like 50 times or more so lipid molecules than protein ones. And if you want to know more stuff about membrane proteins, there's this nice book chapter that you can freely read at the NIH site, this molecular biology of the cell. And I recommend that, as well as the Leninger's biochemistry. And so I will link to this book as well as to various articles that I found helpful, and hope this helped.